So welcome everybody. It's a pleasure to introduce for this first ecotoxicomic webinar Dr. Abdulsamie HANANO, who comes from the Atomic Energy Commission of Syria. And I'm really happy to welcome you for this first event, which is co-organized by the Emerging International Network on Microbial Ecotoxicology, Ecotoxicomic, and the French Foundation for Scientific Cooperation in ecotoxicology. So now let's go with the presentation of Dr. Abdulsamie HANANO.
Okay, good morning, everybody. So before starting my speech, I would like to address a special thanks to Stefan for this amazing invitation and for giving me this interesting opportunity to talk about our research achievements today. So the title of my presentation, as we see here, is Molecular and Biochemical Focus on the Xenobiotic-detoxifying Biosystem, Research, Significance, and Potential Application. Before talking science, I would present myself. So my name is Abdulsamie HANANO. I am plant molecular biologist. I have obtained my PhD from Louis Pasteur University in Strasbourg, France. Actually, I am working as a researcher and a leader group, small group, in the Xenobiotic-detoxifying Biosystem Laboratory at the Department of Molecular Biology and Biotechnology, and more specifically, in the Division of Toxicology and Biochemistry at the Atomic Energy Commission of Syria. So let's start. What is an xenobiotic? Xenobiotic is a foreign chemical substance found within organisms, and normally xenobiotics that are hydrophobic, persistent, and with high ability to bioaccumulate are expected to be more toxic to the organisms. The toxicity could be assessed in microorganisms, plants, animals, and human. For example, in human, the toxicity is related with anomalies or damage in liver enzymes, reproductive organ, and even more in immunity system. They can activate some certain types of cancer and even more cause the death in the case of overdose. So the persistent organic pollutant POPs are the most critical family of the organic xenobiotics. These contaminants are produced by incomplete burning of organic material and go in the chain food from plants to animals and finally to human, where about 95% of contaminants detected in a human body come from the contaminated food. So actually, there is a dramatic increase in this pollutant in our country with potential environmental concerns. So we estimate that there is a urgent need to develop a research project to resolve or minimize this problem by giving a particular attention to the xenobiotic detoxifying biosystem. So as I mentioned before, the POPs are the most persistent organic pollutant that pass and bioaccumulate through the food chain, presented here in this triangle. So our mission was to reduce or to minimize this triangle as much as possible. For that, it is logically important to focus on the starting point where the POPs enter into the ecosystems. I mean by plants or in soil. So our research was focused into parallel projects. The first was to study the effect of organic pollutant on soil microorganisms at the genetic and functional levels, and possibly try to identify some effective biodegrader. The second was to study the relationship between plants and organic contaminants, giving particular attention to the phytotoxicity and other biological response of plants and this will give indication about the possibility to use plants as phytoremediator for this type of contaminants. So we have targeted two types of environmental pollutant in the organic persistent pollutant, the dioxins and petroleum hydrocarbons. Regarding the dioxins, as maybe you know, the dioxins are the most organic pollutant in the environment. And here, just to refresh your memories, this is the chemical structure of a dioxin Tcdd, 2378 tetrachlorodibenzoparadioxin. This is the most toxic congener of a dioxin. Dioxin are colorless, odorless, hydrophobic, persistent, with a proven toxicity. Dioxin are naturally biodegraded by a specific type of enzyme known as dioxygenases, and more specifically named as angular deoxygenases, that have been identified in some species in bacteria like Pseudomonas, Rhodococcus, Sphingomonas. So our first project was to study the effect of dioxin on composition and functions of soil microbial community. The objectives of this project were first to determine the level of contamination in soil sample, then to study the effect of dioxin on structure of microbial community. So soil samples were collected from different regions in our countries known by their heavy industrial activity. So we firstly estimate the dioxin contamination in soil samples, then isolation and identification of soil microorganisms. And this is just to show the detection of a Tcdd by ELISA kit and then analyzes it by Gcms system. So there is some of our results. Regarding the contamination of soil samples with dioxin, it was largely varied, and accordingly they regrouped in 3 levels of contamination, level 1, 2, and 3, from high to low contamination with dioxins. Regarding the composition of bacterial community in soils, apparently the genus Bacillus species was the more abundant in all soil samples, and followed by Arthrobacter and Klebsiella. And we studied the expression of angular deoxygenase alpha subunit. As I mentioned before, this enzyme is involved in biodegradation of dioxin in bacteria by real-time PCR. We found that the expression of this gene is highest in the highest polluted soil samples, as we see here in S9, 10, and 12. However, no AD transcript were detected in dioxin-free soil samples or low contamination with dioxin. So this work was published in the Journal of Chemosphere in 2014. Now regarding the petroleum hydrocarbons. Hydrocarbons compress alkanes, cycloalkanes, and aromatics. Hydrocarbon can contaminate terrestrial environment, but more importantly, aquatic environment, by accidentally spilling or by petrochemical industrial process. So the recovering of petroleum hydrocarbon contaminants from aquatic environment by physicochemical methods is not good for many of reasons. However, the biological decontamination of petroleum hydrocarbon could be a good alternative. Apparently, the yeast are the most active microorganism that can use the petroleum hydrocarbons, and more specifically the N-alkane. And that this was largely studied in some species of yeast such as Candida, Yarrowia, and Pichia. So our second project was to isolate and characterize some microorganisms from petroleum hydrocarbon contaminated soils and go forward to functional characterize of some functional petroleum crude oil degrading enzyme. So we identify a new strain of Saccharomyces cerevisiae at morphological and molecular level. And apparently, this strain of Saccharomyces has a remarkable ability to grow in the presence of petroleum crude oil, where its removal activity was estimated to be about 70% after 21 days. Here, we presented genetic evidence on the presence of a CYP52A3 homolog gene in this strain of Saccharomyces cerevisiae, and apparently the expression of this gene is petroleum crude oil dependent. Moreover, the growth of yeast in the presence of petroleum crude oil was related with significant increase in the CYP proteins content, and more specifically with the induction of a microsomal protein of about 59 kilodalton. Moreover, or more interestingly, apparently the expression of this microsomal CYP is induced by N-alkane and more specifically by N-hexane. So to better identify or better characterize this protein, the full length of the gene was cloned, sequenced, and compared with other identified isoform of the family CYP52A. And as we see here, the protein sequence of our target protein shares a high homology with other protein in the family CYP52A that have already identified in many species of yeast, and it was therefore named by the International Center for CYP as a CYP52A58. Later, we confirmed that recombinant protein CYP52A58 expressed in yeast catalyzes the monoterminal hydroxylation of an alkane with a remarkable preference for those composed of C16. And finally, we suggested this model to explain how the Saccharomyces cerevisiae can uptake the N-alkane from the medium and possibly biotransform them to probably the corresponding fatty acids. So this work was published in 2 papers. The first was in the Journal of Basic Microbiology, and the second was in the Journal of Chemosphere in 2015. Now, another interesting microorganism that I would like to talk about, this is that we have identified directly from petroleum crude oil. It is a new strain of Bacillus safensis. This strain of Bacillus safensis apparently phylogenetically located in the same branch where other strains of Bacillus safensis have been identified. And the ability of this strain to produce biosurfactant was tested by hemolytic activity in A, by oil displacement test in B, and by hydrocarbon overlay plate agar in C. And later, the chemical nature of biosurfactant was characterized as glycolipid. We demonstrated this strain contained a gene named beta-DGS, which is enzyme or coding for enzyme that is required for the biosynthesis of a glycolipid. And apparently, the expression of this gene is induced or strongly induced by low molecular weight of aromatic hydrocarbons, and more specifically by pyrene. So this is a graphic abstract summarized the green story of Bacillus safensis that starts with A and ends with H. From A to E, as I mentioned before, this strain was isolated directly from petroleum crude oil, identified at molecular and morphological levels, and its ability to produce biosurfactant was tested, and the biosurfactant was characterized as glycolipid. And here F shows that the pre-adding of biosurfactant to the liquid medium in the presence of petroleum crude oil and Bacillus safensis that will enhance the ability of bacteria to remove the petroleum crude oil, as we see here in H, a real picture where the petroleum crude oil layer was seriously reduced in the presence of Bacillus safensis. So this work was published in the journal Frontiers in Microbiology in 2017. And now I would like to talk about our second parallel project that we have started 6 years ago. This project based on the understanding of the relationship between plants and organic contaminants. I think it is maybe not very interesting for you as microbiologists to talk about plants, but I think it is important to briefly present or to briefly expose our results in this project. So for that, we use the plant model, Arabidopsis thaliana, treated with a different concentration of the dioxin Tcdd in vitro conditions. And here, I will briefly highlight some of our results. First, apparently Tcdd affect, but a little bit, the seed germination of Arabidopsis thaliana, but more importantly affect the fresh weight of plants treated with the dioxin. So this is the effect of dioxin of the plant morphology and even more in the structure of root system. Apparently, the Tcdd exposed plant have enhanced level of ROX or reactive oxygen species, but without any aspect of cellular death. So apparently plant Arabidopsis thaliana, the plant Arabidopsis thaliana can uptake the dioxin from its environment. And the Tcdd is accumulated mainly in the shoot, but more importantly in the root, and the removal activity of plant to dioxin was estimated to be about 55%. Here, the detection of a Tcdd in a plant tissue by the Gcms, as I mentioned before, it is accumulated mainly in shoot, but more importantly in the root. And more specifically, the Tcdd is mainly accumulated in leaves and seeds of treated plants. And interestingly, and as we see here in the scanning electron microscopic image, the seeds produced by treated plant with the dioxin are small with wrinkled phenotype, and this is apparently related with down or dramatic downregulation of key genes involved in the biosynthesis of fatty acids in a plant. So this work was published firstly in the Journal of Chemosphere in 2014, in BMC Plant Biology in 2015, I think, yeah, and more recently in the Journal of Experimental Botany in 2018. Now I would like to talk about an experiment that we have done in our laboratory. This experiment based on the high affinity between a dioxin and the plant lipid. And this is known by lipophilicity. So our idea was, if we can extract plant lipid as a particle known as oleosome or lipid body and or lipid droplet, to extract or bien uptake the dioxin contaminants from the environment. For that, we use the date palm seeds to extract the lipid body and use them as agent to extract the dioxin from aquatic environment. Why date palm seeds? Because the date palm seeds are considered as a byproduct in the date palm producing country. So firstly, we obtained a pure fraction of lipid droplet from the date palm seeds, as we see here in the light and same image, with intact structure. Then we study the partitioning or the absorption of a Tcdd in the medium into the lipid body, and apparently it is working very well because about 90% of a Tcdd was absorbed by lipid body at pH 7.5. We found also that the partitioning of a Tcdd in the medium into the lipid body extracted from germinated seeds is more important or higher compared of lipid body extracted from non-germinated seeds. So this work was published in the Journal of Frontiers in Plant Science in 2016 in collaboration with Elizabeth Blay from Strasbourg, France, and also in collaboration with Professor Denis Murphy team from University of South Wales in UK. And interestingly, this paper was highlighted by The Economist newspaper under the title "Syrian researcher used dead stones to suck up toxic materials." So you can obtain the full length of article at the indicated link. Now, to finish my presentation, I would like to thank every member of my team, my small team, Muhannad, Ibrahim, in the same order, Mary, and Dua, for their patience. So we are a small team with young scientists, but our ambition is big. Also, I would like to thank my collaborators, Dr. Elizabeth Blay from the Institut de Biologie Moléculaire des Plantes in Strasbourg, France. Also, special thank to Professor Denis Murphy and his team from University of South Wales, UK. Professor Murphy is a leader expert in plant biotechnology and more specifically in plant lipid body biogenesis. So this is our scientific participation to save the planet, and we hope that we will did. However, we need your support to keep our hands beside of yours. So we were still open to all kinds of collaboration. This is my email, and this is my WhatsApp number. And finally, I would like to address a special thank to Fondation Revoltant for giving me this opportunity. And thank you so much for your attention.